All right, guys, let's now take a look at chapter 21. So let me go ahead and share the PowerPoint with you guys. As you can see here, we're looking at the immune system. So I know we just finished talking about the lymphatic system, which talked about different organs and different ways that we can kind of clean the fluid that is being returned back to blood by exposing it to the lymph nodes, filtering it through as it passes through different lymphocytes and macrophages. Here in the immune system, we'll talk about other ways that our body defends itself. So in this chapter, we're going to talk about what's called non-specific immunity and specific immunity. Non-specific immunity, we are, just as that name implies, we're talking about how our body doesn't specifically target just one particular pathogen, what we commonly know as germs. We know that there are different kinds of germs. There are bacteria. There are lots of different kinds of bacteria. There are viruses, and there are lots and lots of different kinds of viruses. And then there's a variety of others. There's different kinds of fungi. There are toxins. There are allergens. I mean, there's all kinds of different things. And our non-specific system is going to defend against all of those things. Anything that's foreign, our non-specific immunity says, nope, you don't belong here. Whereas then once it gets past that, if say it passes that non-specific immunity, then we're going to be very dependent on our immune system, which is then our specific immunity, our ability to recognize things that we've been exposed to before. And again, that's what that is. So our non-specific immunity, we'll talk about how our skin is part of that, inflammation, fever, the process of phagocytosis. Like I said, just anything that's considered foreign is going to be rejected somehow, and our body will do it in these different ways: phagocytosis, inflammation, fever, and some other things. Whereas then specific will differentiate between the specific and non-specific. We'll talk about what's called T cells in what's called cell-mediated immunity, and then we'll talk about the role of B cells in what's called antibody-mediated immunity. Then we'll get into what's called, when we get more specifically into that, so specifically into specific immunity, we'll talk about the difference between genetic immunity, what's sometimes referred to as natural, and then we'll talk about acquired immunity, so immunity that we have to develop on our own over the course of our lifetime through exposure to things. With that, then when we get into acquired immunity, we'll talk about the difference between that acquired. Sometimes acquired immunity happens naturally or artificially. We'll talk about the difference between those. So there's actually 4 different types of acquired immunity, and that's going to be looking at whether it occurs naturally or artificially, and whether it's called active or passive, and I'll explain that. And then we'll get into other immune responses. We finish up with talking about some of our hypersensitive reactions, which would be like allergens and anaphylaxis. And then we'll define the term autoimmunity at the very end of this chapter. So what is our immune system? So our immune system, there are, again, specific and non-specific. Specific immunity targets one specific substance. So meaning the fact that in the last video, I referenced the virus chickenpox. I had chickenpox when I was younger. Because I did, my body was able to develop an immunity for it. Now, if I'm ever exposed to the chickenpox again, my specific immunity can recognize that and knows exactly how to target it because of the exposure I've had before. Whereas non-specific immunity just protects us against many substances. Again, just as that word implies, it's non-specific. So it's not just targeting chickenpox, it's targeting anything, any type of virus, any type of bacteria or toxin of any sort, as it just wants to keep everything that doesn't belong out of our body. So that's what we'll get into first. So we have what's called 3 lines of defense. Just as that, as we're looking at, that means that it has 3 kind of barriers to get through before it can then cause an infection in our body. The first 2 lines of defense are non-specific immunity. The first line would be all of our physical and chemical barriers in our body. So things like our skin. Bacteria cannot necessarily get through our skin unless there is an opening in our skin, or like a cut of some sort, or it gets in through our mouth or our nose or something like that. So skin, but also chemical barriers such as like the mucous membrane inside my nose is designed to trap different debris or different, pathogens so that it doesn't make its way into the body. So, and there's other things with that too. So any type of mechanical or chemical barriers, our second line of defense would be once it passes those barriers, we have white blood cells that we know as phagocytes. Phagocytosis will just target anything that's foreign. We gave the term antigen, if you guys remember talking about that in our blood chapter. Antigen is a synonym for pathogen, which is a synonym for germ. Anything that doesn't belong in our body that we consider as a germ, phagocytes can target that and will try to rid the body of those. So phagocytosis as well as inflammation. If we get a cut in our skin, we may notice that sometimes it gets kind of red around that site, sometimes a little bit swollen, maybe painful, and that is because inflammation is actively taking place to try and get blood to the site to make sure it cleans up any bacteria that may have come in that cut. So we'll talk a little bit about inflammation. Fever, very, very similar. We spike our temperature so that our body can fight off whatever infection is present. Any other type of protective proteins, we'll get into these, and what's called natural killer cells. All of these are all going to target just anything that doesn't belong in our body. Whereas then we'll get into specific immunity, which is our third line of defense. So that is the activation of our B lymphocytes and T lymphocytes, or also known as B cells and T cells. And those particular cells are involved in that immune process, meaning that we are trying to recognize what this pathogen is that has made its way into our body. Have we seen it before? Do we know how to fight against it? If not, we are going to, fight it as best as we can. But in that event that we do fight it, we now develop antibodies in response to it for the next time we are exposed to it. So we'll get into that. So here are those different lines of defense and more of a visual here. So we have our non-specific immunity, as I mentioned, our physical and chemical barriers is the first line up here. So we have like our skin and mucous membranes are those mechanical barriers. The chemical that's, we do have inside our nose, we do have that mucous membrane, but also like inside of our mouth, we have saliva. Inside of the stomach, there's acid that hopefully if we take in any type of chemical that we are eating, hopefully it's destroyed in all of that acid in our stomach. Tears are very much a chemical barrier. They have enzymes in that so that if anything gets into our eye, we try to not just flush it out, but those enzymes within our tears are also going to try and fight against something. Reflexes such as coughing and sneezing are there to try and get things out of our nose and our mouth, as it's it's more of a reflex, so we don't think about it, but that's what the body is doing. Then we get into our second line of defense. So again, the process of phagocytosis, so the ingesting of those foreign materials, inflammation, our body going through fever. We have different proteins called complement proteins and interferons, which we'll get into. And then this is such a goofy picture, but those natural killer cells as well. And so I'll talk to you guys about those. So our second line of defense, so the first line again is, like I mentioned, just our physical and our mechanical and chemical barriers, just saying, hey, nothing that doesn't belong here, we're trying to keep that out. So like I said, our skin, nothing's getting through our skin unless it comes in through an opening. Within that opening, hopefully it then runs into some of those chemical barriers. Like I said, if it comes into our mouth, saliva has enzymes in it to make sure that we are trying to kill off something right away. Leukocytes, what happens is when we have some type of infection, leukocytes will go to that site for infection. What happens in the process of diapedesis, that means that these white blood cells are going to squeeze out of those capillaries and try to get to that site. Chemotaxis is then being able to, there's a picture I think in your all's text that talks about how it's like a bloodhound, able to get out of the door, and then the chemotaxis is then sniffing out that infection, that it's actually able to target that's saying, hey, you don't belong. So those particular white blood cells are now at that site, and those particular those particular white blood cells we know are going to eat. They're going to eat those pathogens, again, the germs that might be present, whatever it might be, whether it's any type of bacteria or virus, whatever it is that doesn't belong, it's trying to get rid of it, as well as any type of cellular debris. So something that may be nearby that site, whether it's an injury site or just an infection. It's trying to rid that area of anything that doesn't belong in that area. So that's the process of that phagocytosis. Hopefully that's something that's familiar to us. Also part of the second line of defense is our inflammatory response. That what happens is, and I think we may have brought this up in our blood chapter as well, that in the event of any type of injury to our skin, then what happens is we sometimes see these classic signs of inflammation. We see redness, heat, swelling, and pain. That's what inflammation is. And that may be because, we have, maybe gotten a splinter, or if we have a cut of some sort, any type of injury to our skin, we sometimes see this because blood is going to rush to that site to try and fight off anything that may have made its way in. Again, any opening in the skin in our body's eyes is not a good thing, because it means now things have the chance to get into our body through a pathway that normally isn't there. So blood is going to rush to that site. So that's what you're seeing with the heat, or with the redness and swelling, and even sometimes the heat, because we're seeing more, everything going to that particular injury site, to try and get platelets in case there needs to be a clot formed at a cut. But also our white blood cells, those leukocytes need to get there so that we can fight off anything that might be present there. So inflammation is oftentimes a good sign, because it means your body is trying to take care of itself. And that's where we're seeing those different signs. Fever is also a good thing. In an extent. So fever, we know as, again, our body temperature spiking. When we see our body going typically above 99 degrees, typically means there's a sign of some type of infection present. So in the process of fever, we have phagocytes that are going to release phagocytosis, the process of, again, that eating of bacteria or virus or whatever it is, releases what's called pyrogens. Pyrogens are simply just fever activating, proteins. These pyrogens, their job is to reset the body's thermostat. We know that, again, our hypothalamus is our body's thermostat that's going to be controlling, our temperature, and wants us to be typically around 98.6 degrees Fahrenheit. Instead, these pyrogens, once they're activated, they spike that body temperature. The body temperature will rise, and what it's doing is that hot temperature. Sometimes pathogens that are present are, feel good at a normal temperature and can thrive and replicate and cause infection. But in the event of a spiked temperature, those pathogens, because of that extreme heat, may then die off. So this is your body's way of trying to kind of cook these pathogens. Sounds crazy, but that's kind of what is happening. So fever is often a good thing. You don't want to lower a fever immediately. After a long period of time, though, and maybe even to a certain temperature, if your child's temperature gets too high, or if they have had a fever for a long period of time, that needs to be brought down at that point. So we have antipyretics, which can lower temperature. So things like Tylenol, ibuprofen, aspirin, those are all things that can lower that fever. So that's why those are there. You can see it on the label. It says that they are pain reliever and fever reducer, and that's exactly what's happening there. But that's what fever does. It is part of that second line of defense, recognizing that something is here that needs to be killed off. So that's what a fever is doing. It's trying to get rid of it by extreme heat. We also have different types of protective proteins. We have what's called interferons, that are proteins that are secreted by virally infected cells. Cells that have been taken over by a virus. The way that a virus works, a virus is a non-living entity. It doesn't replicate on its own. It depends on the host cell. What happens is a virus gets into my body and takes over my healthy cells, and is very dependent on my healthy cells. In order for that virus, it uses my healthy cells against themselves and against me. And so what happens is our body has the ability for those cells that have been taken over by a virus, they have that ability to recognize that they have been taken over. And so what they do is they produce a protein called an interferon that they secrete, and that sends a signal to other cells saying, hey, this has been infected. And sometimes that has the ability to send the signal to a phagocyte. But either way, it protects that cell from viral replication, meaning that these interferons are going to interfere with that virus's ability to replicate and then therefore take over. So it's kind of interesting to think that our cells have that ability to recognize, okay, I've been taken over, I'm probably going to be killed off, so I'm going to secrete this protein that's going to then make sure that the virus doesn't spread to any other healthy cells. So it's kind of our cell's way of helping everyone else out. Then we also have what's called complement proteins they complement the immune system in that they too are very similar. They're just proteins that are circulating. They're inactive normally as they circulate through our bloodstream, but in the event that they're activated, so because of some type of presence of a pathogen, they assist those white blood cells in working in phagocytosis, and they will help to destroy any pathogens that are present. So we do have these proteins that are also something that will help with our white blood cells in our second line of defense. We also have another type of cell that the name implies exactly what they do. They are natural killer cells. They act non-specifically, meaning that they're just looking for anything that doesn't belong, and they are very effective against different types of microbes, which again is pathogens. Microbe is just short for microorganism, like again a bacteria or a virus or what have you, and certain cancer cells as well. But they are just that. They are going to just target that particular pathogen. They will fight it and then kill it off, and that's what their job is. It is a type of lymphocyte, so it is specifically a type of white blood cell. Okay. And then we get into our third line of defense. So our third line of defense, as I mentioned, is now our specific immunity. So you're seeing what's called cell-mediated and antibody-mediated, which we'll get into over here on the side. But what we're looking at here is our specific immunity is specifically targeting a certain kind of organism. Not just, how our non-specific is looking for anything that doesn't belong. Our specific immunity knows how to fight specifically against influenza A, or knows specifically how to fight against streptococcus, a very specific bacteria. It knows how to respond because of exposure to it in the past. And again, if it is the first exposure to a particular type of pathogen, we will probably get sick from it, but our body develops antibodies so that it's more prepared for the second exposure. So let's talk about this. So we have in our specific immunity, we have lymphocytes that are very much involved in this process called T cells and B cells, or sometimes called T lymphocytes or B lymphocytes. T cells are involved in what's called cell-mediated immunity. What they do is these particular T cells are more involved in kind of a cell-to-cell combat, that they will target the pathogen and they will fight it. And that's what our T cells are involved in. We're going to get into a little bit more with that. We'll talk about the different kinds of T cells and what other T cells will do. Then we have B cells. B cells are more involved in the antibody-mediated immunity, meaning that these are the cells that will, in the event that there is a pathogen present, these particular cells are going to work to activate antibodies. So antibodies are the ones that can recognize, or in the event that, again, it's our second or third exposure to something, we're going to produce more antibodies because we've seen this before and we know which antibodies to activate. So that's what our B cells do. They're more involved in that antibody-mediated immunity. So that's how I remember oftentimes the difference between the two is the B reminds me of the B in antibody. So I remember that B cells are more involved in that antibody mediated immunity. And then we have macrophages that are also very much part of that. We talked about macrophages in the last chapter in our lymph nodes. Macrophages are just larger versions of our monocytes, so they are particularly phagocytic cells. But they also help to activate these T cells and B cells. All right, so the term antigen, I've referred to this term numerous times today, but antigen is synonymous with the term pathogen. So any antigen is a substance that stimulates the formation of antibodies. So these are generally large molecules. Most are proteins, but few are, as it mentions, polysaccharides and lipids. As long as you know that an antibody that an antigen is something foreign. That's all you need to know about antigens. Antigens just mean something that doesn't belong. And so in the event that an antigen is in our body, it stimulates the production of antibodies. So let's look at the difference between, again, cell-mediated and antibody-mediated. Cell-mediated very much uses T cells. So T cell activation, what happens is a macrophage, again, is a phagocyte. It will ingest an antigen, so it destroys it, it eats it that way. What happens is the antigen presents it what the macrophage does is it moves the antigen towards its surface. That antigen presentation will then stimulate a cloning of these T cells, and there's different kinds of T cells. We have what's called killer T cells, which do exactly that. They're going to kill off this antigen right away. They're going to attack it. Helper T cells do just that. They actually help to activate more T cells and B cells. Suppressor T cells, that in the event the antigen is then destroyed, we will then suppress the activation of those T cells because we no longer need those T cells anymore since we have destroyed that antigen already. And then memory T cells very much are involved in the activation of those B cells, so using the antibodies. So we're using that memory. So this is going to help with those B cells as well. So B cell activation, so in this antibody-mediated immunity, the macrophage, when the macrophage ingests the antigen and the antigen presents itself, here we're seeing B cells and helper T cells are going to be activated. Remember, we said B cells are what's involved in the production of antibodies. Helper T cells help to produce, in this process to help produce these antibodies and B cells. So we have what's formed. We have plasma cells, which again derive from the plasma, and memory B cells, which are going to be able to remember when we've been exposed to certain things. So there's different types of B cells. The big thing to know here is the difference between, cell-mediated immunity, which is the involvement of those T cells, and then the antibody-mediated immunity is the involvement of those B cells, which will then stimulate, again, the production of more antibodies. So antibodies, again, what I mean by these different antibodies, their job is to destroy antigens, sometimes directly and indirectly. Meaning sometimes an antibody its job is to just directly attack and to destroy these antigens. Sometimes antibodies work a little bit differently. Sometimes they indirectly attack, meaning that sometimes they will attach themselves to an antigen and may not destroy them right away, but it's enough to send a flag or a signal to a nearby phagocyte. And again, those macrophages that are large phagocytic cells, they see that antibody attachment and say, hey, that's foreign, I need to attack that, and the phagocytic cell will come and destroy it. So that's what I mean by indirectly. An antigen, again, is something foreign that stimulates the formation of antibodies. And an antibody secreted by these B cells, oftentimes referred to as also immunoglobulins. It's just a synonym for antibody. What happens in the antigen-antibody interaction, typically the result is what's called agglutination. If you remember, that was the same type of response when somebody with one blood type is given the opposite blood type, which means kind of a clumping that takes place in the blood. And so that's what happens with just the reaction between an antigen and an antibody, and that's how that antigen is destroyed. So here are just a few important certain types of antibodies. Immunoglobulin G is found in plasma and bodily fluids, and is very important and very proficient in attacking bacteria, viruses, and other types of toxins. And that's immunoglobulin G. Whereas immunoglobulin A is found in places like breast milk, is found in our tears. We saw that picture a second ago, and different gastric juices, and it's very effective in protecting against infections in those particular areas. Immunoglobulin M is where we see that, again, I was just talking about the agglutination process with mismatched blood. That's where we're seeing the activation of immunoglobulin M. Immunoglobulin M is found in plasma, and it is involved in that blood compatibility and incompatibility. In the event that the wrong blood type is given, this antibody is what recognizes that. And then immunoglobulin E is very much involved in our hypersensitive reactions, which are things like allergies or anaphylaxis, and we're going to talk about that a little bit later in the chapter. So that's what an antibody is. An antibody is produced in response to an antigen in the event that we were going to try and get rid of it. So why is the secondary response stronger than a primary response? So I've mentioned to you guys before that, again, we will respond better if we've been exposed to something before. Upon the first exposure to some type of antigen, some type of pathogen, we'll call it a bacteria or a virus, whatever, what happens is my body doesn't recognize that. That's the first time we've ever been exposed to it, so we produce a couple antibodies, we may get sick, we fight it off. Okay, cool. The second time we're exposed to it, at this point, my body says, hey, I've seen you before. So because we've fought it off before, we've experienced it, we know which antibodies work best in fighting off this particular pathogen. And so upon the second exposure, what happens is we produce even more antibodies in response to that because we know exactly what it needs, and we produce that in great numbers. And because we produce more numbers, we're seeing statistically we're going to get rid of it a lot faster. So we're seeing that secondary response is going to kill off that antigen a lot faster, and we may or may not even develop any signs or symptoms at all, depending on the particular pathogen. So there's there's 4 different types of immunity. We have genetic, which is sometimes called inborn, and species specific. This is just the immunity that we're born with. It means that, we're born with a lot of our natural immunity, our non-specific like skin and phagocytic cells. So that's how that is occurred, is we're given that. We're passed that on through our genes. But then there's acquired immunity. So acquired immunity means that we have to be exposed to it somehow. So you can see through this little diagram that our immunity can be divided into genetic or inborn, meaning that's just what we're born with. And then there's acquired. And this is where you're really going to develop an excellent immune system by, exposing, if the more we're exposed to something. So we can actually divide this up a little bit further because we're either naturally acquiring that immunity or artificially acquiring that immunity. So you can see down here there's 4 different types. There's 2 types of naturally acquired, and then there's 2 types of artificially acquired. And so the name is just that it's either natural or artificial. So let's look at natural first. So natural acquired is either we acquire it actively. So meaning in the term, anytime you see the term active when it comes to immunity, that just simply means that we ourselves are creating our own antibodies in response to this. So natural active means that we are exposed to the antigen and we produce our own antibodies. So this is going to happen through exposure, for example, from exposure to a virus. So meaning like, I go to school and I sit next to somebody who has the flu and they sneeze on me. More than likely, I'm going to get the flu then. So I am going to get sick. I'm going to maybe be out for a couple of days, but that exposure to the virus, what's going to happen is I am going to produce my own antibodies from this. And so when I go back to school, if I'm exposed to the flu again, as long as it's the same strain of that flu, I won't get sick, or, I maybe won't get as sick or maybe not at all. But that's what I mean by natural active. Whereas natural passive, in this case, the term passive means that we have to be passed on the antibodies. We don't create the antibodies ourselves here. So someone else makes the antibodies here. Something that occurs more naturally is from a mother to an infant. So either through in utero, so meaning in the womb, mother and fetus are exchanging blood. In that case, antibodies are passed on. Or perhaps in breast milk. When an infant gets sick, a mother's breast milk will naturally produce antibodies to help defend against whatever that baby is fighting, as it's, again, a natural occurrence, but it's passed on. The baby is not old enough to be able to fight off that infection right away, so that breast milk will provide that for that individual. So those are your naturally acquired immunities. Whereas artificial can be also acquired either actively or passively. Artificial active, once again, the term active means that we ourselves are creating our own antibodies, but we are artificially passed on these, the ability to do so. So this works through things like vaccines and toxoids. A vaccine is typically a weakened or a killed off version of a virus or pathogen of some sort, like the flu vaccine is a killed off version of the flu, but it is the flu. So when you're injected with a flu vaccine, you are given the flu. Again, it's not going to cause an infection because it is weakened or, again, a killed off version of it, but it still is introducing the flu to our body so that our body can create its own antibodies in response to that. It knows exactly what it looks like. So in the event that I'm exposed to a live version of that exact same virus, I know how to produce the antibodies in response to it. And then that live version would be almost like my second exposure to it, and I'll fight it off before I get sick, or at least really sick. Sometimes people who get the flu vaccine still may get the flu, but at least they don't die from the flu. That's the way to really think about that. So that's what artificial active is. Whereas artificial passive is, again, we are passed on the antibodies. We don't create the antibodies on our own, but artificially being passed on those is we are passed on antibodies that are made by others. So immunoglobulin, meaning that they're just antibodies that are made by others. They have the immunity to it, so it's passed on to us through some type of injection. Things like antitoxins and antivenoms are also an example of this. So say, for example, I get bit by a snake. More than likely, I am not going to be able to fight off that snake venom on my own or quick enough. So an antivenom will be injected into my skin and can fight off that venom a lot faster. Again, I'm not producing anything to fight it off. I am given the antibodies to this. So that's the difference between those four. So going back to that, so these two were natural. We have natural active and natural passive. Natural passive natural active, we are just exposed to that pathogen. We may get sick from it, but we develop our own antibodies. Natural passive is we are naturally passed on those antibodies. So best example would be a mother to an infant through breast milk. Artificial active, again, active meaning that I'm going to create my own antibodies, but that's passed on artificially to me through a vaccine. And artificial passive is I am artificially passed on the antibodies. Okay. So then we'll get into what's called hypersensitive reactions. So we have different types of allergic reactions. It's either immediate or delayed, but allergic reactions are oftentimes called hypersensitive because our immune system is almost responding too much, is a way to think about it. And in the event that we respond too much to it, we might have to counteract that hypersensitive reaction through some type of medication. So we have what's called the delayed type, meaning that we may not immediately see a reaction from this. We see it in about 48 hours. Best example of this would be poison ivy that they mentioned here. The usual case is repeated exposure of skin to different irritants like household detergents or, again, poison ivy. As soon as you touch poison ivy, it doesn't immediately cause a reaction in your skin, but it may later on that night or the next day. And so that's why it's called delayed. What this does is it activates T cells, causing skin eruptions and inflammation, and sometimes referred to as contact dermatitis. And so we see that reaction, and sometimes we have to counteract that with hydrocortisone creams, or again, something that will help take away that rash. But that's what that is. It's a hypersensitive allergic reaction. In some allergic reactions, we have the more immediate type, which is your more common type of allergies, such as bee stings, allergies, food allergies like peanut allergies or lactose allergies, or even just pollen and seasonal allergies. In a more severe case, that gets into anaphylaxis, which it means cuts off of your bronchial passageway and can therefore inhibit your breathing. What happens in this type of immediate reaction, an allergen will activate a B cell. The activated B cell forms a clone of the antibody secreting plasma cell. The plasma cell secretes large amounts. Remember, immunoglobulin E is the last immunoglobulin that we discussed on that antibodies page that is more involved in this process. IgE will then bind to the mast cells in body tissues, and therefore more allergen invades the body. So you can kind of see this process taking place in this visual here, that B cells activation of B cells by the allergen will produce plasma cells and memory cells, which will, cause a secretion of those particular types of antibodies, which bind to different mast cells and then therefore release histamine. And histamine in our body, if you remember, is involved in this process to try and stop that allergen. But it is more of a hypersensitive reaction, and because we release so much of it, sometimes that can cause bronchial constriction, so constricting of our airways, as well as a large quantity of vasodilation, meaning that more blood is rushing through our body, can cause a lot of issues. So that's why oftentimes in the event of an allergic reaction, in more mild allergic reactions, we can take things like Zyrtec and Allegra and those types of over-the-counter medications that will help kind of counteract that histamine and that allergic response. Or in the event of some type of anaphylactic response, then we see more extreme cases where you have to carry an EpiPen. And again, that large dose of epinephrine will hopefully then open up those bronchial passageways and eventually kind of calm our body down. And that's how those allergic reaction works. And those work. And those are referred to again as hypersensitive reactions. Other types of immunological responses, we have what's called autoimmune diseases. There are lots of different examples of this. Lupus, rheumatoid arthritis is an example of this. But in this case, what happens is normally our body is supposed to recognize the difference between self and non-self. Self cells are normal. You belong there. We are supposed to destroy the non-self, like bacteria. But somebody who is experiencing autoimmune disease has a problem with recognizing the difference between the two and instead attacks everything. So people with autoimmune diseases, their healthy cells are attacking themselves. So that's where we see self-attack, or what's called autoimmunity. And that's what that is. It's a type of immunological response. That's not always a good one. And there's also organ rejection too. Recipients' immune systems will recognize a donated organ as something as foreign, and sometimes will mount an immune attack against it. Somebody who has some type of organ transplant, who receives an organ from another individual, such as a kidney or a liver transplant, will typically have to be on some type of immunosuppressant for the rest of their lives in order to hold back that type of response to it, to say, hey, this is okay, please don't reject this organ. As that helps with that process. So that's another type of response, is again, our body typically wants to fight off something that isn't normally there. All right, guys, so that is the end of chapter 21. So again, you can see how those two chapters went together, our lymphatic system and our immune system. So make sure you look over those. We've got one more chapter to cover this week, and that is our respiratory system. So it doesn't really have anything to do with our lymphatic system, but it's usually a good one to cover with these two. So go ahead and move on to that video when you get a chance.